Welcome, students of Standard 10th. Let's begin with a chapter, Genetics. Genetics is totally a new concept to you children. Before explaining or before going in detail, let me define what is genetics. Genetics is the branch of biology which deals with the transmission of features, differences and similarities from parents to offspring. Now, who is the father of genetics? Father of genetics is Gregor Johann Mendel. He is considered as the father of genetics because he worked or he conducted various experiments on hybridization, and he formulated a theory called Mendelism. Now, what is heredity? Heredity is the process of transmission of genetically based characteristics from parents to offspring. Heredity is based on the phenomenon called like begets like. It is called as like begets like. It means young ones look like their own parents. We resemble our parents, yet there are differences between the children of same parents. There are differences in the individuals belonging to the same species. Now, these differences are called as variations. Variations are caused due to crossing over. Now, what is crossing over? Crossing over is defined as the phenomenon in which the genetic materials are exchanged between the maternal and paternal chromatids of homologous chromosome, which results in the formation of new combination of genes. And due to this new combination of genes, there are differences, there are variations. So Gregor Johann Mendel, he conducted various experiments on hybridization, and for his experiment he selected a plant called sweet pea plant. The botanical name of sweet pea plant is Pisum sativa. To know more details about Gregor Johann Mendel and the advantages or the reasons of selecting plant, let's focus to the explanatory parts. An Australian monk who had great interest in hybridization in plants, Gregor Johann Mendel. He performed certain experiments on hybridization for about 7 years, and he selected, for this experiment, he selected a garden sweet pea plant. He selected this plant for various reasons. Now, what are the reasons or what are the advantages of selecting this plant? First, many varieties were available having contrasting traits. Varieties were available in pure forms. Peas are normally self-pollinated because pea flower is bisexual. Self-pollination could be prevented by removing corresponding reproductive parts of the flower. Cross-pollination could be done artificially because the size of the flower is very convenient. And the last point is reproductive span of pea plant is very small and 2 crops can be produced in 1 year. Now, before going to the explanation of various laws formulated by Gregor Johann Mendel, let us be clear about the biological terms related to this chapter. So, we will come now to the terminology used in heredity. Now, what are genes? Genes are the hereditary units present on the chromosome. These are physical, discrete, definite, stable bodies that carry characters from parents to offsprings. Allele. An individual in which the factor or the allele is the alternative form of gene. This is very important, children. You have to mention it is the alternative form of gene occupying same position on homologous chromosome and affecting the same characteristics in two alternative ways. Now, next, homozygous. What do you mean by homozygous, or it is in the pure form? An individual in which both the factors or genes controlling one character are similar. Once again I am repeating, an individual in which both the factors or genes controlling one character are similar. For example, now capital T, twice it is written. So, here both the factors controlling one character means height are similar. So, it is pure tall. Now, here also both the factors or genes controlling one character are similar. Now, small t represents dwarf. In heterozygous condition, you will come across an individual in which the two factors or genes controlling one character are dissimilar. For example, capital T, small t, it is hybrid tall. Here the two factors are dissimilar. One is dominant and other one is recessive. So, it comes under hybrid tall, whereas in this case the two factors or genes are similar. So, it comes under pure tall. Similarly, hybrid round, capital R stands for round shaped seed which is dominating, and small r stands for wrinkled seed which is recessive. Now, next we will come to dominant allele. In heterozygous condition, the allele which expresses itself morphologically, means externally or physically, is called dominant. Recessive, in heterozygous condition, the allele which does not express itself physically and remains concealed or unexpressed is called recessive. Now, next, genotype. Genotype means it is the genetic constitution. Genetic constitution means, for example, you look here. Now, the genotype, when it is asked in the question, write the genotype factor, or genotype means you have to mention for the pure tall capital T twice. This is the genetic constitution. Hybrid tall, capital T and small t, this is hybrid tall, and for the dwarf, small t twice. So, genetic constitution of an individual for any character due to which it expresses itself is called genotype. Phenotype. Phenotype is the morphological expression. It means appears to be. External appearance, or you can say morphological expression of any character of an individual is called phenotype. For example, tallness, dwarfness, red color flower, white color flower. Next, we will come to the monohybrid cross. Monohybrid cross, a cross between two individuals having one pair of contrasting traits, a cross between two individuals having one pair of contrasting traits producing hybrid generation is called as monohybrid cross. For example, a pure tall plant when it is crossed with a dwarf. So here pure tall plant is crossed with a dwarf. So the gametes are here from the parent, the gametes are capital T for the pure tall and for the dwarf is the small t. On combination of the gametes, we get genotype. So in the offspring which is produced in F1 generation in monohybrid cross, the genotype or the genetic constitution is capital T and the small t. So here in F1 generation, the offspring which is produced is hybrid tall. So it is hybrid tall, the offspring which is produced by crossing the pure tall plant with a dwarf plant is hybrid tall in F1 generation. Next we will come to the dihybrid cross, a cross between two individuals having two pair of contrasting traits producing a generation in which all individuals are heterozygous for both the characters. Once again, a cross between two individuals having two pair of contrasting traits. So here in dihybrid, we have to take two pair of contrasting traits, whereas in monohybrid we have taken only one pair of contrasting trait. That is, one pair means only one character was shown taking as height of the plant. So here we have to take two pair of contrasting traits producing a generation in which all individuals are heterozygous for both the characters. For example, pure yellow, this is the color of the seed, pure yellow and round seed, when it is crossed with green colored and wrinkled seed. Now pure yellow is dominating, so capital Y twice you have to write down, round twice you have to represent, and green is recessive, small y, and for wrinkled, small r. Now the gametes for this capital Y represents yellow colored seed and capital R represents round shaped seed. Similarly, small y represents green colored and small r wrinkled. So on combination of the gametes, the genotypes formed here, capital Y, small y, capital R, small r. So in F1 generation, the individuals or the offsprings which are produced are yellow and round seed. So the offspring which is produced in F1 generation, the plants have yellow colored seed, yellow and round shaped seed. So here yellow is dominating, round is dominating. Though recessive factors are there, but the dominating factors appears morphologically or externally. So the genotype of the offspring produced in F1 generation during dihybrid cross is yellow and round shaped seed. Now next we'll come to the 7 pairs of contrasting characters. Mendel took different varieties of plants showing 7 pairs of contrasting characters. Now what are the 7 characters and the contrasting traits? So the 7 characters are here. Plant height. So tall is dominant and it is represented as capital T twice, and the dwarf is the recessive factor, it is represented as small t twice. Similarly, when you take the color of the flower, purple is dominating, white is recessive. Color of the pod is here is the fruit, green is dominating, yellow is recessive. Dominating means which expresses morphologically. If you take color of the seed, just reverse to the color of the pod. If you take color of the seed, yellow is dominating, green is recessive. Shape of the seed, round is dominating, wrinkled is recessive. Then again shape of the pod, inflated is dominating, constricted is the recessive. And if you take the position of a flower, axial is dominating and terminal is recessive. So these are the 7 different characteristic features or 7 pairs of contrasting characters found in Pisum sativum. So he formulated 3 laws. Mendel formulated 3 laws. Now what are the 3 laws? First law is the law of dominance, law of segregation, law of independent assortment. Now let me explain what is law of dominance first. Now law of dominance states that in heterozygous condition, you have to mention this, in heterozygous condition, out of the 2 contrasting alleles, one expresses itself morphologically and other remain unexpressed or suppressed. The factor or allele which expresses itself, we call it as dominant. The factor or allele which remains unexpressed is called recessive. So here one example, again I am going to repeat. Now a male plant which is pure tall is crossed with a female plant which is dwarf. Now what is your observation here, children? You see, the offsprings which are produced here in F1 generation are all hybrid tall, because pure tall when it is crossed with a dwarf. So the offsprings produced in F1 generation, these offsprings are hybrid tall. Next, based on this, I am going to show Punnett square. Now Punnett square is a checkerboard where it shows the combination of the gametes to form genotypes. It is a checkerboard where it shows the combination of gametes to form genotypes. Now in the form of Punnett square, how will you represent the genotypes? Now this is male, female. After the combination you will get here capital T, small t. So here the 4 genotypes. Now you see 4 genotypes, that means the 4 offsprings are hybrid tall. So when it is asked to write the phenotype, you have to write that the offsprings or the individuals formed in F1 generation are hybrid tall. So when it is asked to write down about the genotype, here you have to write down the genetic constitution of the offspring, that is capital T and small t. So this is about F1 generation. Now in F1 generation the offspring is hybrid tall. In F2 generation, that is on selfing, the offspring produced in F1 generation, now we'll see the genotype of F2 generation. So on selfing, the gametes, that is male and female gametes on selfing it, so the genotypes which are produced in F2 generation are so here what you see. Now in this case you are seeing that the genetic constitution of all the 4 individuals are same. But when you come to the F2 generation, you see there are variations, there are differences in the genotype as well as in the phenotype. So when you go to the phenotype, that means appears to be. So when you go to phenotype, appears to be, now this is pure tall, these two are hybrid tall, and this is dwarf. So here 3 are tall, appears to be tall. So when it is asked to write in ratio, so it is 3:1. So 3 appears to be tall and 1 is dwarf. 3 appears to be tall and 1 is dwarf. So the ratio is 3:1. Phenotypic ratio is 3:1 in F2 generation of monohybrid cross. When you go to genotypic ratio, you have to specify very clearly pure tall, is 1, hybrid tall, that is capital T small t, 2, and pure dwarf, that is small t, twice 1. So the genotypic ratio is 1:2:1, and the phenotypic ratio is 3:1, and the genotypic ratio is 1:2:1 in F2 generation. Now I'll come to the law of segregation. Now what is law of segregation? Law of segregation states that the 2 factors or the genes controlling one character separate. Segregation means separation. Separate or segregate without blending or influencing each other during the formation of gametes, so that each gamete receives one factor or gene for each character. So this is very interesting. So due to this reason, law of segregation is also known as law of purity of gametes. During the separation, the gametes, though they separate, but they will not mix without blending or influencing each other during the formation. So that each gamete receives one factor or gene for each character. So it is also called as law of purity of gametes. Now children, I am coming to the conclusion of genetics part 1. Gregor Johann Mendel convinced that it will not be long before the whole world acknowledges the results of his work. So in genetics part 2, I will be teaching about dihybrid cross, then the Punnett square will be shown based on dihybrid cross, and the law which verifies the dihybrid cross is the law of independent assortment. Now next, this is the self-evaluation test. I have given some questions, please work it define gene, allele, genome, dominance, homozygous condition, heterozygous condition, differences between gene and genome, gene and allele, monohybrid and dihybrid cross, haploid and diploid. Then next, I had given one question I have given based on this, few bits are there. A pure tall plant which is represented as capital T twice is crossed with a pure dwarf plant, small t twice. So, you have to draw Punnett square to show F1 generation, F2 generation, give the phenotype of F2 generation, give the phenotypic and genotypic ratio of F2 generation. Thank you, children. So, next class I will be teaching about the dihybrid cross and about the diseases, hereditary diseases, that is X-linked inheritance, Y-linked inheritance, about the mutation. Under mutation it comes gene mutation and chromosome mutation. Thank you, children, and have a good day.